We have a wonderful program, and I get to introduce our sort of star speaker to introduce the program, and that's Cori Bargmann. And you have a nice summary of all the incredibly wonderful things she's done, but she actually, I met her at one of the HDF workshops. And those HDF workshops were really unique because unlike anything else I ever participated in, there were geneticists who were working on little C. elegans. That's an elegant worm that's found in the soil. And she could put together all the little neural networks in that worm. And that would tell us much about the mechanisms that happen in our much, much more complex networks in the brain. And at any rate, we all reviewed grants together, and Nancy and her father always organized these wonderful dinners, and we learned from each other, and it was really wonderful. And so I think we can tell that she did her postdoc with Robert Horvitz, Nobel Prize winner. And then I guess that wasn't enough for her because after she first was at the University of California, San Francisco from 1995 to 2004, but then came to Rockefeller and married a Nobel Prize winner himself, Richard Axel. And now in 2016, she was appointed the president of science at the Chan Zuckerberg Initiative. So that's an incredible, impressive bio. And so I can't wait to have your words of wisdom for us. So welcome.
So hello, everyone. It's an honor for me to be here with you today. As Anne says, I'm Cori Bargmann. I'm a geneticist and I'm a neuroscientist. And usually when I get up in front of an audience of people, I talk to them about how genes and the environment cooperate to build neural circuits that give rise to behavior. And I talk about things like how motivational states are generated and how the brain interacts with experience to cause changes in the brain and changes in learning and in outcomes, and even evolutionary changes in the way that genes encode behavior over time. And usually at the end of a talk like that, there will be a typical moment where you stop and you acknowledge the people in your lab who actually did the work. You'll say, oh, here's a student who did all these brilliant imaging experiments I told you about, and here's a postdoc who did a lot of great analysis or genetics. And then after that, you thank the funding organization that gave you the resources that made it possible to do that work. But more and more over the past few years, when I've thought about my own work, I've realized that there were a lot of people who were really critical and really important for making work move forward, who weren't on the list of people who were being thanked. And those were the people who were developing resources and technology, and those were my scientific collaborators from different fields who were teaching me different fields to allow science in my group to move faster. So to give an idea, everything that my lab did for many, many years, and still does, is built on a resource that was developed in the 1970s and 1980s by a man named John White working in the UK, who put together a complete wiring diagram from head to tail of the organism that I study. And 30 years later, that's still the resource I turn to every day. And another thing was that the Genome Project of C. elegans was done as an early project in the 1990s, and the person who did that, John Sulston, went on to be one of the major players in the Human Genome Project as well. And I would never have put John Sulston as an author on my papers, and I wouldn't necessarily put him on my acknowledgement slides. And yet everything that my lab did in genetics and understanding what the genes were that allowed neural connections to form and neurons to migrate long distance to find their partners, was building on the foundation that he had built for human genetics. And there are many other examples of times that a tool that someone spent a decade or more developing suddenly made something possible in my group. And other examples of times when I would work with people, for example, in theoretical or computational neuroscience or population genetics, and they would teach me things that I didn't know, and we would come up with something together that was better than any of us could have done alone. And that's the framework that sort of gratitude and of reflection that was on my mind when I met Mark Zuckerberg and Priscilla Chan about 4 years ago. Now, on the birth of their first child, Max, Mark, the founder of Facebook, and Priscilla, his wife, decided to give away 99% of their wealth in their own lifetime to help to build a better future for everyone. And their thinking about this has been inspired by, in many ways, Priscilla's own experiences. She is the child of immigrants, the first in her family to go to college. She was a teacher after she finished college, and then she went to medical school, and she's now a practicing pediatrician. And so her great interests are in opportunity and justice, things like immigration and criminal justice reform, in education and the opportunities that that gives everyone to do their best, and in biomedical science. Because as a pediatrician, she says, so many children are children that you cannot help because there is no help. In order to work with them day after day, you need to have hope. And science, she says, is hope. And she wants to support science that will provide hope, not necessarily for herself, but for her children's generation and going forward. So these three initiatives, science, education, and justice and opportunity, are the initiatives that the Chan-Zuckerberg initiative is built around. And all of them are coming to it with a bit of a slant from Mark Zuckerberg about thinking about technology and computer science and data and the various kinds of tools that can be used now to make lots of different kinds of parts of life, including the nonprofit side, work better. So when I met Mark and Priscilla, I asked them what they wanted to do with this tremendous gift that they were making for science and other topics. And they said, well, we want to support science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. And, you know, the idea of solving all diseases is kind of a mouthful, but just to put that in context, I want you to think about where we were in medicine 100 years ago and what science has done for medicine since then. Because realistically, about half of the things that people were dying from 100 years ago, science has made it possible now to cure, prevent, or manage. We have antibiotics to treat bacterial diseases. We have blood pressure drugs and statins to prevent heart diseases. We understand that things like cigarette smoking cause cancer, and therefore we can prevent those diseases as well. And even in my own lifetime, I've seen remarkable progress on diseases that seemed intractable. So in work that I started when I was a graduate student at MIT working on cancer, and that led to a whole host of people working together over a decade at MIT and at UCLA and at Genentech and other places, a very basic science discovery about a gene that could mutate to cause cancers in rats led to the development of a rational cancer therapy, Herceptin, that is used to treat women with a very aggressive form of breast cancer. So a basic science discovery about a rat cancer is a step along the path with hundreds of other steps toward human treatment. In the same way, in my own lifetime, I've seen AIDS go from a disease where the diagnosis was a death sentence within a year to a disease that is now manageable, where people are living for decades based on antivirals and understanding of the disease that moved the science forward and moved the medicine forward. And most recently, Frank Bennett is here with us today. I can remember the first experiments where people said, you know, something about making antisense RNAs seems to be able to shut genes off. And that's led just in the past couple of years to the development of an RNA-based therapy, the first sort of disease-modifying therapy for a human neurodegenerative disease, spinal muscular atrophy, that can radically help these children that otherwise would not be able to really ever be able to sit up or walk, to now live successful lives. I was at a ceremony last year at the Breakthrough Prize where one of the children who had been treated with these drugs that Sam had made was up on the stage, and I can tell you there wasn't a dry eye in the house. The idea that this could move forward from science is something that everyone embraced. So I really believe that science can move medicine forward. You are not going to be able to talk me out of this. And the great thing about Mark and Priscilla is that they recognize something else about science, which is that it's really hard work and it's really slow. So they're not saying solve all diseases in a decade or in 2 decades. They're saying 100 years. What does it take to make progress in 100 years? And I think that's because they're young people and they want a better future for their children as well as themselves. And they understand what it will take to make that happen. Now, even all of that said, 100 years is pretty aggressive. So I thought about what they were saying. I thought, you know, maybe 500 years. And Mark said, no, technology can make anything happen faster. Mark is an engineer. And so we thought about that, and we came up with what we thought was the right approach, which is, instead of trying to name 11,000 diseases and cure them one at a time, let's try to recognize that science is really powerful and that scientists are incredibly motivated, and let's try to help them by accelerating their work. Let's turn to the scientific community for the discoveries we want to make, and let's make every scientist a better scientist. Let's make science go faster. And here are theories about how we can make science grow faster. And they're really, for me, based very much on my own experience in science. And I can say that they're also inspired in part by the work that the Hereditary Disease Foundation has done. I first started interacting with the HDF just after I started as an assistant professor. I was on the board from 1992 to 1997. And I saw certain things that influenced the way that we think about not just one disease, but many diseases. So for example, we think collaboration is really important. We think that the problems that are out there now are complicated problems, and we need people to work together with different expertise to solve those. And we're trying to do that in part by bringing new people into fields, which was something that the Hereditary Disease Foundation did very early on. They would bring people from fields of genetics, model organism genetics, into the study of human disease. They would bring people who had never thought about this somewhat obscure neurological problem into the room to start to think about how they could use the powerful tools of human genetics to gain insight into it. And this collaboration was really always a part of what they did. We're inspired by them, actually. One of our starting projects is to work on neurodegenerative diseases. Again, with the idea that we want to think big, we're trying to think of approaches that would be helpful for thinking about the several hundred neurodegenerative diseases, of which Huntington's disease is one. But again, we're thinking hard. What does it take to bring new people into a field? How do we bring new ideas in? Maybe we should have some immunologists. Maybe there's something about the immune system in these diseases. Well, we for sure need some doctors to tell us what they are if we're going to be bringing new people in. And setting up those kinds of relationships and those kinds of virtual institutes is something that the Huntington's disease group was doing decades ago that we're trying to imitate. Second idea we have is that technology is really important, that every scientist is as good as his or her tools, and that better tools make things possible. The Genome Project is a great example of a technology that lifted all boats. And because it was a general technology where every disease has some sorts of genetic components or risks, and therefore knowing what all the genes are moves forward research on every disease. So with that spirit, the Chan Zuckerberg Initiative is working extensively on a project called the Human Cell Atlas, which we think of as something like the Genome Project, only for cells. So everyone knows that cells are the fundamental unit of life. Humans have about 25,000 genes. Humans have about 37 trillion cells. That's a lot. But fortunately, they're not all different from each other. They fall in categories. But actually, we don't have a parts list of the human body yet. We don't know how many different cell types there are and exactly where they are and what genes are active in each cell type. And yet, every disease has a cellular basis. Every disease represents some set of cells that are malfunctioning in one way or another. And Huntington's disease begins when the medium spiny neurons of the striatum start to malfunction. They're a special class of neurons. They have special properties. Knowing about a cell in a dish or even a different kind of neuron doesn't tell you what you want to know to really move this forward. And so again, here's a case where we think, if we can really put together that whole parts list, we can help people make progress against every disease. We can help every scientist use that as a foundation for their future work. What else? We think people need to share information, not just with their immediate collaborators, but also more broadly across science. We want people to build on each other's work. We've been supporting things like the preprint movement that started at Cold Spring Harbor that enables scientists to share their work when they're first ready to share it, maybe when it's still a little rough around the edges, but lets other people build on the work more quickly. And we're also trying to help people discover each other's work more quickly using tools like machine learning and artificial intelligence to help point people toward things that they might not know about that are relevant to their research through a project that we're doing that just went live last month called Zuckerberg Meta. That's a way of finding new ways of interacting with the scientific literature. So these are all ideas that the Hereditary Disease Foundation has worked with in its own past and continues to work with today. It's always been about eliminating the different separations between fields that kept doctors and scientists from talking to each other. It's always been about encouraging collaboration, particularly in the human genetics at an early point when that was not the practice. It's always been about interdisciplinarity. They brought new ideas into the fields. They supported technologies and resources like tissue banks for people to share. And they focused on open science in these meetings that were in sort of unexpected and intimate settings, and that really had a teaching and learning attitude. Like I remember at the beginning of every meeting, Anne would get up and draw the circuits of the basal ganglia so that everyone could see which arrow went to which arrow and which were which pathways that did things. And you know, frankly, it probably was like 4 or 5 times before I could have drawn it myself. So it was a useful thing. It was bringing everyone up to speed in a new field and a new topic. Now, this is very much the work of one extraordinary family, Milton Wexler, Nancy Wexler, and I want to turn now to Alice Wexler, who wrote a book about the Hereditary Disease Foundation and the work they did together. Just a quote here on how her father, Milton Wexler, after his wife's suffering from Huntington's disease, began to work on this problem. Alice says, "My father's deepest commitment was basic research. At a time when the disease was of interest primarily to a few neurologists and geneticists, my father and sister helped create a support system, seed money, tissue banks, pedigrees, and workshops that enticed many basic scientists to study Huntington's. In doing so, they pioneered imaginative ways of working with scientists and of fostering dialogue among scientists, which would have implications far beyond the Huntington's research community, a community they helped create." And I just want to say that those implications continue in the work that we're doing in the Zuckerberg initiative, because they brought more than collaboration and technology and open science. They brought and they understood the importance of people. And Milton Wexler was a psychiatrist, and it was his job to understand people. And he thought about how to motivate people. And you ask yourself, what motivates a scientist? And scientists are really motivated by discovery, by curiosity and the discovery of learning new things. And they're motivated by respect, and they're motivated by having autonomy and the ability to really do things on their own. And really, scientists are motivated by the desire to make an impact. And what the Hereditary Disease Foundation did was help them see how that was possible. So in addition to bringing in a mix of scientists that was quite broad and a lot of fun, And celebrities, movie stars, famous architects, people that a scientist would normally not run into. Most importantly, what the Hereditary Disease Foundation did was to bring patients to those workshops, was to bring the patients into the room and treat them with the respect that they deserved. And, there's patients brought to the scientists the answer to the question, why am I doing this? Because one of the hardest things about being a scientist is that you wake up in the morning and you move transparent liquids from one tube to another tube, and sometimes you're just not really sure why you bother. And by having patients in the room, you think, this is why I'm doing this. I want to be able to make this difference. They bring the inspiration, they bring the urgency. And the presence of the patients made barriers melt away, and they made egos melt away, because all of a sudden that seemed more important than whatever else was going on. And so more science happened because of that presence, because of that human engagement. And then something else happened, which is that better ideas emerged because the patients were part of the conversation. That it wasn't just the neurologists describing their symptoms to them. It was them bringing up what they thought the important questions were. Disease is the experience of a human being, not the observation of a specialist. And Alice Wexler, again, in her book, highlights this as one of the very special things the Hereditary Disease Foundation did to move science forward. Huntington's is also about non-scientists playing an active role in science, not only through fundraising and lobbying, but by participating in decision-making about which research to support and working with scientists to organize research efforts. By intervening directly in the scientific world, these Huntington's activists significantly influence the priorities and practices of biomedical research. Moving patients from positions as research subjects to research participants to really drivers of research is something that the Hereditary Disease Foundation pioneered. And I should say, in AIDS, the AIDS activists are another really stellar group that had that role in bringing science forward. And patients can ask the hard questions, like, who does this research belong to really? They're an inspiration to us. We at the Chan Zuckerberg Initiative are really interested in expanding that inspiring model. And with us here tonight is Tanya Simoncelli, who is the leader of our science policy program. 2.5 years ago, I met Tanya at a AAAS meeting in Washington, sat for coffee, and realized that I had to work with this woman one way or another. And many of you may know of Tanya, even if you haven't met Tanya before, and that is because she is tied up inextricably with a really important story in human genetics, which is who owns human genes. Tanya started at the American Civil Liberties Union in 2003 as its very first science fellow. Science had not been something that organization, which thought about themselves as being about human rights and civil rights, was really concerned with. But Tanya started there, and she started learning about the areas of science that could influence civil liberties, and she was struck by the fact that a company, Myriad Genetics, had patented the human gene where mutations in that gene gave rise to breast cancer, BRCA1. And she was really electrified and galvanized by this and believed that this was violating a fundamental human right, the right to know about your own genetics in your own body. And starting around 2005, she started convincing other people at the ACLU that this was a stance that they should be taking, that they should be fighting for the freedom of human genes. And she framed it not as a patent, an incorrectly issued patent, but really as a civil rights case. You have a right to the use of your genes. So Tanya, with the ACLU, filed a case against Myriad Genetics, and were accompanied, just really inspired and rallied around them, a large number of other people, including patients, for example, who hadn't been able to afford tests for breast cancer or hadn't been able to have better tests for breast cancer because Myriad, with its monopoly position, had gotten a little lazy about finding new mutations. And it included doctors like Wendy Chung, who's here with us tonight, who recognized the importance of this for their own patients and moving medicine forward. They filed the case in 2009 in the Southern District of New York. They won. They lost on appeal. They went all the way to the Supreme Court. And in the Supreme Court, they condensed their case to 4 words: are human genes patentable? And the Supreme Court in 2013 answered no in a landmark 9-0 decision where they agreed that the human genes belong to humanity, belong to us. And one person, Tanya Simoncelli, really drove this case from its inception to its conclusion in inclusion. So Tanya has been in other places, including the FDA and the Office of Science and Technology Policy, and we're incredibly fortunate that 2 years ago she joined us as our first Director of Science Policy. And I just want to say that her policy work is very much inspired by and builds on the work that the Hereditary Disease Foundation has done, its approaches and goals. Can we make patients the drivers of research? We have just started a program called the Rare as One program that will help patient groups for other rare diseases build up the kinds of support that the Hereditary Disease Foundation has provided for Huntington's disease. And spearheading this group of patient groups and finding out ways of building these groups forward is the heart of Tanya's program. Hundreds of groups applied to this program. Sadly, we're only going to be able to support about 10% of them in the first round. But we want to use this cohort to build tools, build expertise, and create a community of practices that can advance progress against many diseases. Now, it won't be true that every disease has a Milton Wexler or a Nancy Wexler or a foundation of the luster of the Hereditary Disease Foundation, but you've proved that it can be done, and it should be done more. You are our inspiration. And you are also a piece of work. And I mean that, of course, in the way that Shakespeare says it. What a piece of work is man, or in Nancy's case, woman. How noble in reason, how infinite in faculty, in form and moving, how express and admirable, in action, how like an angel, in apprehension, how like a god, the beauty of the world, the paragon of animals. Nancy, you are a piece of work. We hope to all be a piece of work. Thank you. Chan Zuckerberg Initiative, people, collaboration, technology, open science, and the patient as a driver of research. Wish us luck.